Today we're going to focus on first summarizing what we talked about on Tuesday, and then we're going to talk about one final problem, which 72% of you already know what that problem is. We have an end replication problem in eukaryotes that prokaryotic cells don't have. So we'll talk briefly about that and how we deal with it. And then we're going to move back into our biological and chemical warfare agents, both mustard gas and anthrax, and how they relate to DNA replication. Excuse me. All right, so let's put it all together and review what we went through on Tuesday. So number one event in DNA replication is an initiator protein. Again, in prokaryotes, that's now DNA A, right? And in eukaryotes, that's ORC, the ORC complex, okay? And may, see that big word, may separate the strands at the replication origin. DNA A will separate the strands, it will melt the strands and open up, providing accessibility for DNA B to bind, the helicase to bind, right? In the case of ORC, ORC does not melt the DNA strands and relies on accessory proteins for that function. Yes?
Wait, so can DNA B not bind, but not separate them by itself, and that's why it needs to be
It needs...
because it can't separate at like the initial, or?
No, I don't know that that's entirely the case, and I don't want to say that that is the case. In the bacterial system or in prokaryotes, DNA A will partially melt, right?
Yeah.
It will immediately partially melt the strands.
Yeah.
Whether or not, I would guess that that's a requirement for DNA B to clamp on and start working, but I don't know if that's the case. In other words, if I had another mechanism to partially melt, right, a region of bacterial DNA, would DNA B bind and start a replication fork? Perhaps. I don't know. I don't know that answer. Yeah. Yeah. Yes. Yes. Yeah. So I think that's a great way to think about it, right? The helicase is going to use energy, right, to break those hydrogen bonds. In the case of DNA A, there's something structurally about that protein that when it binds is able to break those initial hydrogen bonds, right? But it's an initiator. It can't. It's not going to proceed. It has no, right? We talk about processivity of the DNase, right? Or the DNA polymerase, right? And helicase also has that processivity because it has that ring structure that fits over the single strand of DNA, and DNA A does not have that. Okay, all right. Okay, so helicase, again, DNA B or the MCM complex is the helicase. It's going to further separate the strands at the replication fork, and it's going to continue to do so throughout the process, as opposed to the initiator protein that just gets things started. Okay? All right, we have this protein gyrase, again, that's going to bind itself around the double-stranded DNA. And I thought there were some great questions that came up both on Piazza as well as right before class. Gyrase has endonuclease activity, right? It's going to actually be able to clip double-stranded DNA, right, in the middle of the strand, meaning not at the end, which would be an exonuclease. So it has endonuclease activity. It also has other activities which allow the unwinding portion of that protein, okay? All right. Primase is an RNA polymerase. It's going to use the DNA as a template to create a short RNA primer, because remember, DNA polymerase 3 can only extend a strand. It can't create a strand. So something has to exist, and it can extend that based on a template. Okay, so DNA polymerase is going to recognize that RNA primer, and it's going to begin to synthesize DNA in, again, a 5' to 3' direction. All right? We're going to have, DNA polymerase is going to generate on the leading strand one long stretch of DNA, and on the lagging strand, primase is going to continue to make short RNA primers that allow DNA polymerase to bind and to extend those RNA primers, again, in the opposite direction, creating what we call Okazaki fragments, right? Okazaki fragments are the RNA primer plus that little bit of DNA that's extended up until it hits the next RNA primer, right? And it won't proceed. DNA polymerase 1 is the DNA polymerase that's going to help us clean up those RNA primers. It's got that exonuclease activity, so it recognizes the end of that RNA primer and it clips it and it replaces it with a DNA. And then it clips the next one, replaces it with a DNA. And so what we then do is, instead of generating a gap, a bond gap between the RNA primer and the original strand, now we have a gap between the DNA polymerase 1 replacement of that RNA primer and the original DNA fragment. And now we're going to have an enzyme called ligase, right? Ligase is the last guy that comes in and he repairs that little nick in the new DNA sequence, okay? And that's going to happen both, obviously, multiple times on the lagging strand and once on the leading strand, right? Because there's that one initial RNA primer, okay? Any questions about that? Okay. All right, so we have an end replication problem, and some of you know that. I'm impressed, right? Okay, so linear chromosomes are going to wind up being shortened through every single replication cycle, right? So if we have our little cartoon up here again, and we have that RNA polymerase, that primase, comes in, it synthesizes the little RNA primer. Pol 3 comes on. Well, I shouldn't say Pol 3 because it's a eukaryotic only. So DNA polymerase delta, right, comes in, extends. This is an error in this cartoon, right? We're getting into specifics, and now we're talking about where there are breaks and non-breaks in DNA. This should be contiguous right here. So it's going to extend that. So there's no break in the... So DNA polymerase 3 is going to, let's say, extend. Let's say that's the leading strand, right? It's going to replicate it. So there's nowhere now for DNA polymerase alpha to bind. DNA polymerase alpha binds double-stranded DNA, and it recognizes that RNA primer and has exonuclease activity to cleave it. But there's nothing over here for DNA polymerase 1 or alpha to bind to, right? So what's going to happen is that RNA primer is going to decay, and now we've shortened that one strand, right? One of the daughter strands is an RNA primer shorter than the parent strand. Let's say that's the entire chromosome, so another round of replication, we look at it from the other side. We generate an RNA primer, DNA polymerase 3. Again, there's nowhere for DNA polymerase 1/alpha, right? So alpha to bind. Can't replace the RNA primer, and we keep shortening every single replication. You can imagine this is a kind of a problem, right? So how many times are our cells replicating even just during development, right? I'm a lot bigger than when I started out as a single cell, right? And so the ends of these linear chromosomes, they have a specific name. They're called telomeres. And these are explicitly a region on the ends of linear chromosomes that are defined by a repeating subunit, okay? So how we deal with this, telomerase is this enzyme called telomerase, and it is a DNA polymerase that carries with it its own RNA primer, okay? So here's the DNA polymerase. That's the protein component. Here's the RNA primer. It holds within it. And as you can imagine, it has the same RNA primer. And so you're going to get this repeating subunit. So it's going to base pair match, AT, GC, or CG. And then it's going to now fill in or extend this DNA strand, right? And if you notice, it's not a palindrome, but it has a repeating unit here, AAC, on the end. So it's always going to generate an end strand of DNA, these last three bases, TTG. So it can always bind it. So you're going to get this repeating sequence of TTG, GGG, TTG, GGG, TTG. You all right? Does that make sense? Yes? No? Yeah. Yeah. Correct. So what this is doing is effectively we've shortened the chromosomes, and this enzyme is then re-lengthening them, right? So telomerase is trying to build this zone, this repeating unit. It's trying to build it in one direction, and replication is trying to eliminate it, right? And so it's the actions, the sort of complementary actions. Yes? Yes. Okay. Everybody understand this? Okay. So, yeah, in the back. Mm-hmm. Yeah, it's so I think you have it exactly right? So at the end, our original chromosomes have this sequence, have many repeats of this sequence of TTG, GGG, TTG, right? That's the repeating sequence. And then TTG, right? Or no, excuse me. So it'll have let's think about that. It'll have a TTG, GGG, TTG, and then that's what it's using to bind, and it's extending that. So then it'll just be this repeating unit of TTG, GGG, TTG, GGG, TTG, GGG. So all it's doing is it's trying to extend the DNA. And if you leave it to run, right, and you make the conditions optimal, it will extend that. If it has enough reagent, right, enough DNTPs and enough energy, it will just continue to synthesize DNA in that sequence, right? And that's its job, is just to lengthen our chromosomes. Right? Yes?
So the cartoon only shows the telomerase working on one of the strands.
Mm-hmm.
How does the other strand fit in?
Yeah, so what's happening is I can extend the DNA. Let's say here's the critical endpoint, but telomerase is extending it out. Now I can have primase, right, the RNA polymerase, now create a primer that's downstream, and now DNA polymerase delta, right, can now synthesize that DNA strand. You're still going to have this asymmetry, right? So if you look here, we're always going to have this weird asymmetry in our chromosomes. We always have this overhang that's based on the length of that RNA primer, the initial RNA primer. Okay? This is our way of counteracting that effect over time. Yes?
How does the RNA
Get into the enzyme?
Yeah.
It's that's a great question. I would suspect it's synthesized that way, right? That there's an RNA somewhere in the processing of that protein. It's sort of loaded with an RNA primer, right? Yeah. Correct one. It's the same. It's the exact same RNA primer. There's no other telomerase primer, right? Does that make sense? Yeah. Okay. Right. Because the protein is actually what's holding that primer. So there's some level of recognition of the protein also for that specific primer sequence. Does that make sense? Because it's holding it into the protein, right? Sort of like, well, we'll talk about it, right, a little bit later when we talk about translation of proteins. It's a similar mechanism. Yeah. Yeah, so no. Right, it's critically not a translationally active region of the chromosomes. It's virtually nonsense, right? We'd get these repeating subunit, right? You'd have a tumor, right? A dimer, right? But a repeating protein of 2 amino acids, right? And so this is a non-coding region of the chromosomes, but we still are going to wind up having erosion of these telomeres, right? So if you look at the length of your telomeres when you're born versus the length of your telomeres today versus the length of your telomeres as a senior citizen, they are shortening, right? They are shortening throughout your life. Part of it is a function of telomerase not keeping up with replication, right? Another is loss of activity of telomerase over time, right, within your cells, that the protein activity just is not as active. And so telomerase erosion in culture, in cell culture, leads to this thing we call cell senescence, okay? And what that means is that at some critical point, they've eroded to a place where now the cell can no longer undergo cell division, right? Either the critical kinases that get us over the hump to undergo cell division are not activated, or things are inhibited, or likely both. Yeah.
I'm just curious, are nucleotides found like free floating in the nucleus, or are they more common for like what would prevent the nucleotide
So yes, you have DNTPs that are free floating, right? Can they base pair? Sure, but there's nothing to stabilize them, right? So you need the enzymatic activity to form the phosphodiester bond, right? And so sure, they can bind and unbind, but that rate is probably fairly high, right? Yep.
Here's a thought question, but if you wanted to theoretically like make the telomerase like keep up with that replication process, could you potentially elongate it twice?
Yeah, so that's a very interesting question, and I will tell you that there have been people that have been working on that. So it was, I think, maybe 7 or 8. I'm going to show my age. It might have been more like 10 years ago. It feels like just yesterday. This was, I think this even maybe made Time Magazine or something like that, and it was the fountain of youth protein, telomerase, right? Which was if you could extend your telomeres indefinitely, that you would never age, right? That you could immortalize a cell, and you can, in fact. In a cell, in culture, if I jack up telomerase activity, then I can have that cell divide forever, right? I can immortalize that cell. The belief is, so you can think, right? You're kind of playing with fire though. So one of the things that it's believed that telomeres protect against are things like cancers. So if you start having a highly dividing cell and telomerase can't keep up with the rate of replication, then all of a sudden there comes a point in time when there are natural mechanisms that arrest that cell's ability to undergo cell division, right? So it's a protective mechanism. It also means, but you'll find a lot in biology that the relationship between regeneration, which is the field I'm in, and cancer are really close. Like we want one but not the other, but we don't know how to get one but not the other. And we also want to live forever, but in living forever, all the methods would likely generate lots of tumors in it. So you might live forever, but as a tumorous mass, right? So pick your poison. All right. Yep. So that's a great question. So someone asked, is it happening during replication? It is happening during replication. I need to look into whether, I don't know, do you know, is it happening even during non-synthesis phase of mitosis? I think it's only during replication. It does what? That's the implication, is that telomerase activity over your lifespan is somehow losing activity. Yeah. Yeah. Okay. All right. Yeah, you had another question.
Quick question.
Yeah.
So since you get these with exponential telomeres, theoretically the more replicate, like more times the cell replicated, the more peaks you get in the telomere. So theoretically, is that also a technique of looking at these telomeres?
Yeah, so as it happens in reality is the opposite happens, right? Is that the telomeres don't actually do a great job of keeping up with replication. And so, but can you use it as a dating system? So in a relative sense, my guess would be yes, right? So if I were to look at your telomere length, I could tell whether or not unless you've got some other things that are unexplained going on, based on the length, I can tell whether you're older or younger, right? But I don't know that biological systems in general are sufficient in this particular case to say, oh, your telomere lengths are, a 1,000-mer. That means you are 17 and a half years old, right? I don't know that it has that level of fidelity. But I'm sure there are people looking into whether or not you can date. But it's interesting from a standpoint perhaps of dating fossils, right? Or trying to get a sense of like how young or old something is, if the DNA is recoverable. Be interesting. Anybody else? Yeah.
Going back to that diagram that we had, is there a responsible rate only responsible for creating one extra nucleus, or can it go either longer?
As long as the enzyme is active, right? The only thing that's driving it is the DNA polymerase activity that it has and that primer. So as long as the primer can find a complement, right, to bind to, and as long as the DNA polymerase has activity, it's just going to keep synthesizing, right? Yep. And there's also, you can imagine, a stoichiometry associated with it, right? So it's going to undergo base pairing, it's going to extend, it is going to fall off, it's going to have to rebind. So the more telomerase, right? So if I am expressing more protein, I have a stronger probability of another telomerase binding and extending, right? And so whether or not aging is a function of loss of telomerase activity, it's probably, if I had to guess, more of a function of loss of expression of telomerase, right? Or loss of efficient production of telomerase enzyme, right? Okay. All righty. Okay, so let's move into anthrax and mustard gas, and then we're going to use this to sort of dovetail very quickly into mechanisms that we have to repair DNA damage, and then we're going to talk a little bit about a pretty cool biotechnological tool that's been hot over the last about 5 to 7 years or so. So the pathogenesis quickly of anthrax is that, again, remember you're going to breathe, the real pathogenesis of anthrax is when you breathe it in. In a spore form, right, is this environmentally resistant form of the bacterium that's in a latency phase. Macrophages will phagocytose that spore, and then the spore is going to come out of that latency phase, right, out of that dormant phase and begin to germinate, to proliferate, and then exhibit some kind of cytolysis, which is essentially that the bugs divide until the cell pops, literally just pops, right? And so that's why you get that necrotic core, that nasty looking black blister that I showed in the beginning of the lecture. All right. So it's going to escape, and that is really where the toxicity of anthrax is in sort of this macrophage killing, and then now once it's out and it's proliferating, it can begin to invade other cells, and you just get this massive necrotic mess. All right, so we're going to introduce this concept of antimicrobials. Everyone here is probably very familiar with antibiotics. Antibiotics are a subclass of this broader class of molecules known as antimicrobials. Antibiotics are specifically synthesized by microorganisms against other microorganisms. So this would be things like fungi that get infected by bacteria and they develop mechanisms, right? These are defense mechanisms for other microorganisms against one another. So we can discover those, right? And we can purify them, and then we can commercialize them, and then give them to folks. Antimicrobial is a broader term that includes both these natural products, antibiotics are natural, as well as synthetic substrates. And so they may or may not, these synthetic substrates may or may not be inspired by antibiotics, but nonetheless are sort of usually organic chemistry products. All right, so the desirable properties, obviously we want selective toxicity. I'd like to kill the microbe, but not you if I'm giving you antibiotics, or I want them to be stable, cheap, easy to administer. I want them to be bactericidal, not bacteriostatic. So bacteriostatic is simply stopping, halting the bacteria, right? And what this allows the bacterium to do is to begin to evolve evasion mechanisms from your bacteriostat, right? So now all of a sudden I can get the bacteria through evolutionary processes can now become resistant to that bacteriostat. So what I want to do is I actually want to kill the cell, explicitly kill the cell. And again, this relates to this term here. Up here, the microbe should not become easily resistant. Again, if we are delivering bacteriostats, that could be a problem. All right, obviously we want to get it to the right tissue distribution. If I've got an infection in my lung, I don't want the antimicrobial to go to my toe. That would not really help. No adverse reactions like allergies, and an appropriate spectrum of activity. And what we mean by spectrum activity is how many microorganisms is this antibiotic or antimicrobial going to affect, okay? So as you can imagine, broad spectrum means I'm impacting a large swath, many different species of bacteria, okay? Versus a narrow spectrum. So I have an antibiotic or an antimicrobial that is specific, highly specific for a specific type of bacterium, right? Like anthrax, right? Okay, so the problem with broad spectrum antibiotics, your doctors like to give them to you because they're easy, is anybody have a Z-Pack? Everybody take a Z-Pack when you're sick or something, got a nasal infection? Here's a Z-Pack. It is a broad spectrum antibiotic. The problem is that with broad spectrum antibiotics, and you don't need to be a hypochondriac, it's okay to take a Z-Pack, but you do have to be careful, is that they're non-discriminatory. So they're attacking both the pathogenic bacteria, but also your commensal populations, right? So your gut is teeming, your lung is not as teeming, but it still has these commensal bacterial populations that are actually helping regulate things like inflammation in your body and are in the gut really sort of helping with breakdown of foods and performing a lot of symbiotic functions. It's important that the ecology of your gut stay in balance, right? And so the analogy here is what happens if we went to, the Shenandoah and killed all the predators, right? All the wolves, bears, yes, we do have mountain lions, snakes, all of that. Now all of a sudden, we have a bigger problem, which is that there's no natural predators for deer or bunnies or mice or rats. And all of a sudden, they wipe out all the vegetation. So now you've got a desert because you've got nothing that's keeping these other species in check. So that's the analogy here, right? Is if I kill everything, then what can happen is I can get an opportunistic bacteria that, something like C. difficile, that's a real serious clinical problem, that can now just bloom in your body and cause real problems, okay? And it's interesting, there's a whole area of microbiome research that's going on that shows that if you take a broad spectrum antibiotic and I begin to monitor your bacterial populations in your gut, they never recover to the original state, ever. What emerges on the back end is completely different than what you started with. Now, it may still be symbiotic, but it's different than where you were. Right? So, all righty. Antimicrobials, there's different modes of activity. We can inhibit cell wall, expression of cell wall. We can inhibit things that are impacting, disrupting cell membrane function, protein synthesis, any part of the life cycle and the necessary functions of a bacteria. Obviously, if we can inhibit them, then we can usually kill that bacteria. So, CRISPR. CRISPR is the best therapy for anthrax. It is one of the most powerful antimicrobials available, though some bacteria are starting to emerge as resistant. It's the only effective treatment of inhalation anthrax, and it is this type of molecule known as a fluoroquinolone. And it's specifically going to inhibit bacterial DNA gyrase. So that tells you right there, when I tell you the mechanism of action is that, then you should all be thinking, oh, I know where this thing screws up a bacteria, right? Where? I heard it. I heard a whisper of it. Supercoiling, right? So during DNA replication, so the bacteria's got to divide, it's got to replicate its DNA, it's got those replication forks, but it has no gyrase, right, in order to cleave and unwind that supercoiling. And so, as you can imagine, what happens is we get this overwound region of DNA, and it physically stalls the replication forks, which will eventually lead to things like DNA breaks, which then will lead to cell death. All right, mustard gas, I'm not going to touch on this much. It's not really known how it acts as this vesicant. Again, this blister forming. Yeah, you had a question?
My question was regarding, so if you apply that CRISPR, does it have to be before the cell lysis occurs, or can it be after? And like, how many days that protein will be?
Oh, that's a great question. I don't have the answer to. I don't know what the window of opportunity is, right? Because just like anything, you do have to get out in front of the bacterial infection. And if it gets to a certain degree, I'm not sure there's enough CRISPR to really rescue that process. But I don't know what the critical time constant is on that. Probably a few days at max, right? In fact, I think it's probably about 2 days. Yeah, something like that. Okay. Okay. So, however, if we look at mustard gas, it has a pronounced effect on DNA, primarily chemically modifying guanines. Okay, so if we go back and look at the structure of mustard gas, it is this alkylating agent. Remember, this chloride is a good leaving group, and it will couple with these purine bases, and you can get everything from alkylation of one of these bases to full-on cross-linking of two guanine bases. Okay? All right, so how does that affect replication? There are a number of different impacts. So one is if I have two guanines that are side by side, I can form what's known as an intra-strand, so within the same strand, intra-strand cross-link, and then this is going to significantly inhibit DNA polymerase from being able to, for complementary base pairing, right, to occur, right? And so you'll get stalling of DNA polymerase in those regions. Same with alkylation, so we might not have two adjacent guanines, but I can still add this bulky alkyl group here, right, that's dangling off that guanine. And again, I'm unable to get effective base pairing there, and we'll get stalling of DNA polymerase. Where the real problem comes are in these interstrand crosslinks, right? So now we've got not just hydrogen bonding holding the two strands of DNA together, we have a covalent bond that's holding those two DNA strands together. Helicase is trucking along, trying to melt DNA, and it hits one of these spots, and it'll stall, number one. And number two is then, over time, helicase will actually generate an entire strand break. So it'll actually break the DNA right here, okay? All right, so stalled replication forks and strand breaks. All right, so you can imagine then that, yes, go ahead.
Going back to the inner strand, like the that bond right
Mhm.
there, would that also cause supercoiling if it's not able to use it, or would you simply be like helicase can't break through?
The helicase can't break through. If anything, you could imagine if it just stayed there, if it just completely stalled, you wouldn't get any more supercoiling, right? Because supercoiling is a function in the case of replication of pulling those strands apart, right? It's a direct response to that.
So why does the helicase actually break the strand? Like, why does it change its function?
It won't, it's not changing its function. Its function is simply to melt, right? And that is a rigid bond. And so it's, you know, it's the thing of the weakest link is what happens, and it just winds up breaking the DNA at that point of rigidity, right? Yeah. It's just it's only got that one job, to melt. All right. So, so interestingly, mustard gas and derivatives of mustard gas have actually been used as a chemotherapy. And why this would be is that the theory was that if I have rapidly dividing cells versus your normal cells, which should be not as rapidly replicating, that cancer cells would be more sensitive to this kind of agent than your normal cells. Now, if you go to the hospital now, they will not give you mustard gas as a chemotherapy, I promise. All right. So, but this is a nice dovetail into naturally occurring DNA damage and how our cells would try to overcome something like this. So in the case of mustard gas, we get a double-stranded break, but there are other kinds of base damage. The first that we'll talk about is this process known as depurination. So it's going to be effectively the hydrolysis of this glycosidic bond, the bond between the base and the sugar, and you get this depurination. And now, if I don't have that base, I have nothing to base pair against, right? So I've lost all information regarding what was at that base. So that's a significant problem. Number two is this process known as deamination. This is a process that occurs on cytosines, and it's going to be basically the removal of this amine group to yield uracil. So we're going to convert cytosine into uracil. So now, when we undergo complementary base pairing, instead of having guanine as our complement, now we have thymidine, right? Yeah.
That wouldn't change, that doesn't happen on DNA. That wouldn't change the ribose sugar in the deoxyribose.
Correct. Yep.
That uracil on its deoxyribose.
Yeah. Okay. All right. So interestingly, I know that Dr. James talked about sort of hypermutations that are occurring during the creation of antibody variants. And this is actually one of the processes that occurs naturally in those cells to derive diversity within that. All right, so then the final one of base damage is the use of ultraviolet light, and ultraviolet light can generate these thymidine dimers to form. So again, similarly, it becomes very difficult for proper base pairing to occur. All right, so how we're going to repair these, we have a specialized set of enzymes known as repair endonucleases. Endo, again, cutting in the middle of a strand. And these repair endonucleases are going to recognize errors. Again, we got a question on Piazza about how endonucleases or any of these enzymes recognize errors. Again, the best thought is through physical distortions in the helix, either non-tight bonding, so you might get a little bit of a base that wants to bubble out. I talked a little bit on Tuesday about wiggling, right? If it's not really well, if it's not forming hydrogen bonds really well, then there might be a certain level of thermal fluctuation going on with its position. And these enzymes likely have a mechanism in order to detect that. All right, so what they're going to do is they're going to recognize these errors, and then they're going to use their endonuclease activity to simply cleave out that damaged base. So it cleaves out the damaged base. DNA polymerase comes down. It can sit on double-stranded DNA, right? It's already got a strand. It's primed by a strand here. It can extend it depending on how much is cut out here. And then again, DNA ligase is going to come in and seal, right, fix that final little nick in the DNA. Okay? All right, when we are dealing with double-stranded breaks, so this might be ionizing radiation. In that case, we get a double-stranded break, and there are two different mechanisms that our cells will use in order to repair those errors. The ideal scenario is using this process known as homologous recombination. And so what's going to happen is we're going to get exonucleases that bind to this damaged end and start nicking off the edges of these, right? So we're going to start trimming these things off. And then what's going to happen is this process where it begins to, I hate using like sentient words on biochemical processes because they're not really looking for, but in this way I'll use it. They will begin to look for the second allele, right? So you have two copies of all your genes, right? And so what it's going to do is it's going to look for the complement allele on the other on your other chromosome. And so what it'll do is it'll use that. You can see here, the second allele will undergo this process known as strand invasion. And so it's going to invade here into this damaged section. And then DNA polymerase will use the second strand on that second allele to synthesize a DNA strand. Right? So it's going to use the second allele as the template to repair that big chunk that it's cleaved out. And so then DNA polymerase will use that repaired version in order to generate the repair. DNA ligase will come in here, clean up, this end, these two ends. And as you can see, the repair has a little bit of that second allele. So it's got a little bit of now diversity in it because it's using the second allele as a template. All right? So it's not ideal, right? It's not perfect, but it happens to be the more robust way to repair these injuries. The less idealized mechanism is this process known as non-homologous end joining. Again, this is a process that occurred during the antibody, right? The splicing of those various regions of antibodies, V, E, and J, or something like that. Is that right? VDJ, right? Yeah, thanks. The VDJs, that undergoes this process of non-homologous end joining. And so what's going to happen is you get this double-stranded break, and you're going to get basically clipping of the DNA sequence on either end, and also addition of bases, complementary bases, that then allow DNA ligase to seal these. But the problem is that that's a really non-specific mechanism, and so you can imagine if I get non-homologous end joining, but I've got additional bases that have either been deleted, so I'm missing parts of the original gene, or I've had some that are adding, then we'll talk about things known as open reading frames. You can actually frame shift your codons, and now you can get non-functional proteins. So if this happens in the middle of a critical protein, now all of a sudden you don't make the protein and things like cancer start to emerge. Right? Any questions about this? Okay. All right. So the final slide is this description of CRISPR. And so CRISPR, the reason we're bringing it up, it's a really hot topic, was discovered actually a while ago, but only realized from a biotechnological perspective over the last, again, 5 to 7 years, maybe a little bit longer than that. And essentially, it is using this process of homologous recombination in order to introduce designed or engineered introductions into your genome, or using non-homologous enjoining methods in order to disrupt protein expression. All right, so CRISPR-Cas, so Cas9 is an RNA-dependent nuclease. It will cleave, it has 2 endonuclease domains, this RUBC and HNH domains. And this protein then couples with this CRISPR strand. That CRISPR strand couples with this, I think it's called a tracer strand. And what happens is this little leader sequence right here, this RNA leader will wind up giving us, using complementary base pairing to target the endonuclease activity for very specific regions. And so CRISPR comes from, I wrote it down, it's a nasty word. It is clustered, regularly interspaced, short palindromic repeats. Whew. I will not ask you that on the exam. And so this is actually an interesting enzyme that evolved in bacteria for host defense. So this is the way bacteria will defend themselves against viral infection. So these sequences bind to viral DNA, and they use endonuclease activity to simply cleave the viral DNA in order to prevent the growth of that virus within those bacteria. And so what we've done then is, what folks have figured out smartly is you can actually synthesize what's known as a single guide RNA, this sgRNA, which has a targeting sequence that is complementary to some region of your gene that you want to perturb. Okay? So let's say there's ongoing work to fix your CFTR. We talked about cystic fibrosis and CFTR, right? There's the vast majority of that disease is based on a point mutation at amino acid 508. And so one could envision targeting endonuclease activity to the CFTR gene, having it bind, and those endonucleases clip, and now I have a double-stranded break. So now my cell has an option. The cell can either undergo non-homologous enjoining. Now, if I want to create a knockout, right? If I want to knock out a protein in order to study its impact scientifically on development of the eye or the heart or the brain or something like that, then I can generate this kind of product that's going to cleave right in the middle of that specific gene, perhaps in one of the exons, right? One of the regions that's going to actually be encoding protein. And now when I get this really messy process of non-homologous enjoining, I start introducing mistakes in the middle of that exon. And more likely than not, when we introduce errors into the middle of an exon, you wind up disrupting protein expression. Or the protein gets expressed, but it's not properly folded, and so then it gets targeted for degradation via a process known as ubiquination. Or what I can do is I can trick the cell into thinking that it has an allele, right? Here I've created an engineered second allele that has some homology with the two ends of the cut, but has this designed, engineered introduction of a gene that I want, right? So one thing that we do a lot of is I can target, let's say, a very specific protein, but I want to monitor it in a developing animal, and the only way I can do that is with microscopy. Well, now I can actually try to introduce a fluorescent protein at the end of that specific protein. So now I get a protein fusion that has that protein X, that has some function that I'm interested in, and now I can look at the fluorescent fusion component that I've added via this type of technology, right? And now I can monitor that protein throughout its function. So this is just, I'll end there, but a really cool technology. A lot of people are exploring it as a potential for both gene deletion, but also this process known as gene editing. I won't say genetic engineering, that's a little bit different, but gene editing is really where this fits. And so a couple folks are into DNA replication, mostly in the departments of biochemistry and molecular genetics, but I saw this come up today, and so I thought I'd throw it up there. Maybe you can see it. Here's an assistant professor in chemistry and molecular genetics. He's actually going to give a talk in a couple weeks on editing, regulating, and imaging the genome using CRISPR technology. So if you're interested, this is over in the Battle Building, which is the Children's Hospital. And if you have the time, it is on Friday after our exam from 12 to 1.